Fantastic. So my name is Rian Pierneef, and I'm going to start off by saying I'm a bioinformatician, so you will not find me in the field or in the lab. And today I'll be talking a bit about the work I've been doing on microbes, microbiomes, whole genome sequencing, and all of that relating to big data and how research on microorganisms has actually become very big data. So the talk will start off with a bit of an introduction. I generally like to give a brief history of next generation sequencing, how bioinformatics and computational biology comes into this, then look at some of the research projects I'm involved in, and then I'll go on to public data and how important I think public data is and how actually accessible or available it is, and then we'll conclude. So I think it's always very important to realize the history of life. And I love this picture because this to me really emphasizes how old the world is and how much of life and biology is out there. And I mean, that is what we are looking at. We are looking at life and biology. So if we were to take Earth's history and compress it into one calendar year, 365 days, this is what it would look like. So on the 1st of January, Earth is born 1 minute, 1 second past midnight. So in January and February, we want to stay inside because of this incessant wind and rain. But then round about February 25, we see life appearing. March 28th, we've got photosynthesis, and then across August and September, we see multicellular organisms and sexual reproduction. November, fungi, fish, land plants, and insects appear, and in December, amphibians and reptiles. And on the last day of the year, at 24 minutes to midnight, we humans appear. So if you look at Earth's history as a calendar year, we've only been here for the last 24 minutes of the year. And then 1 minute to midnight on the last day of the year, we start agriculture. But what this really just illustrates is how much evolution there has been and how much of life we are investigating when we're looking at microbes and microorganisms. So generally, we like to think ourselves as being on top of the tree, but in reality, we only a very small portion of this tree of life. And what we are interested in is actually looking at all of this other branches of the tree of life, the bacteria and the archaea. And this actually consists the majority of life out there are the microorganisms. So let's take a look at the history of next generation sequencing, because this is the technology we use to produce the data which we analyze and how we look at the living world. So in about 1977 or in 1977, Frederick Sanger started the Sanger method. And then this quickly evolved up to the 1990s, where the Human Genome Project was launched. This led to various advances and increases in technologies and capabilities of sequencing. And in 2008, the Human Microbiome Project was launched. Currently, we're in the era of third generation or long generation sequencing. And all of this has evolved to enable us to digitize biology, to convert biological information into digital information. If we have a look at the genomes and the organisms that have been sequenced and cataloged, it started off with a partial tRNA of yeast, and it quickly expanded as the sequencing technologies evolved. Around here, we've got the early Sanger sequencing technologies, which moved into next generation sequencing around the 2000s. And currently we're here in the blue section, which is the third generation sequencing technology. So let's have a look at the cost of sequencing. Now, as with any technology, the more we use it, the cheaper it becomes. So the ability to produce vast amounts of data have become cheaper and cheaper and cheaper. And currently we are sequencing a human genome for less than $1,000. So this then leads to the fact that we are generating copious amounts of data, which needs to be analyzed, needs to be stored because of the cost of sequencing going down. So what I want you to realize is that in essence, when you do perform sequencing, what you're doing is you're producing data. And what you are paying for is data. So every time you sequence something, you are actually paying for the production of data. Now, this is an example of what I would be paying for mobile data on my phone if I were to download movies and do anything on my phone. And that, in essence, is what NGS or next generation sequencing is. It's the production of data. Now, looking at microbiomes and environments, some of these environments require more data and as such more money to be completely sequenced. I've had a look at some marine samples, and the amount of data we need to reconstruct genomes from marine samples is a lot less than what we need for soil samples. Soil samples are a lot more diverse. So what one would need to keep in mind is that the complexity or the diversity is equal to the amount of money you need to pay or the amount of data you need to produce, in essence, to get a holistic picture of what it is you're looking at. So how does next generation sequencing generally work? So in our field, we are looking at microbiomes, and here we can use metabarcoding or metagenomics. I am also interested in whole genome sequencing, where we would be sequencing from an isolate. So the steps would generally be you take your sample of interest, you extract your DNA, and then you can do either metabarcoding, where we target certain regions. We can sequence the whole genome or the full genome, or we can do metagenomics, where we're sequencing all of the organisms or all of the life present inside the sample. So these are the general steps that you would do, is you would take your specimen. For whole genome sequencing, you would grow your pure cultures, do your DNA extraction, some quality control. We do library preparation, and then we throw it on our sequencing machine, and then we produce raw data. And again, this is what the whole process of sequencing is about, is producing raw data. Because this is focused on microbiomes and metagenomes, the steps are the same, but what we have is we've got a mixed microbial community. We extract the DNA, and then we can either do targeted sequencing, where we're looking at 16S or RTS regions, or we can sequence all of the fragments, all of the DNA. Now, this is all a bit boring, but in essence, what we're actually just doing is we're taking DNA from a sample, and we're taking very sharp scissors, and we're cutting up these pieces or fragments of DNA. And then what we need to do is we need to put them back together, similar to building a puzzle. So in essence, next generation sequencing is reconstructing a puzzle from lots of different pieces. Now, this allows us then to build puzzles on our organisms of interest. This would be for whole genome sequencing, and I'll show you a bit on the metagenome or microbiome sequencing in the following slides. Now, what we need to keep in mind is that next generation sequencing produces a lot of data, and that is lots of big puzzles. So what I've got here is an example of the different sizes of some of these genomes and how much data we actually require to completely sequence a genome. Again, if you're looking at soil samples, the amount of data you need to produce to fully reconstruct the organisms present in this is a lot. And as such, what we're doing is we're actually building very, very big puzzles. So in my opinion, next generation sequencing is the digitization of biology. We take biology and we convert it into a computer readable format that we are able to store, analyze, and work with using the latest technologies. So I'm a bioinformatician, and what I work with then in essence is the data. Now, bioinformatics and computational biology has various definitions, where it's the science of collecting and analyzing biological data, or it's the collection and classification of biological information using computers, or it's a branch of information science which is concerned with large databases and information. Now, I think it's all of this and then some added. So in essence, what we do in bioinformatics and computational biology is we take digitized information, like this FASTQ file on the left, and we try and reconstruct genomes that we are able to place it along the tree of life, that we understand where these samples originate and these organisms come from, to get us a better understanding of life and where everything fits in. So I'm going to start off by talking a bit about microbial genomics, because I think this is an important part of microbiomes and metagenomics. It was actually the basis of a lot of the work where we sequenced and analyzed whole genomes in an isolated form. Now, this isn't always possible for all of the organisms out there, but this did lead to a greater understanding of what is happening in the world. So here we're building individual puzzles of individual organisms. So I started off my research looking at publicly available data. And the aim was to scan all of the publicly available bacterial genomes and have a look at the genomic islands present in these genomes. Luckily enough for me, these genomes were all available on the NCBI, and I could go download it right from there and then use bioinformatic tools to scan all these genomes and look for genomic islands and events of horizontal gene transfer. This enabled us as well to have a look at the evolution of certain genera. Using publicly available data, we were able to download vast amounts of genomes and have a look at segments of these genomes to indicate the evolution of, in this case, the Jew Bacillus pangenome. And again, all of this based on publicly available data without me having to go into the lab and do any of the lab work myself. Now, this led to various projects where we were looking at the reconstruction of whole genomes and then depositing these genomes into publicly accessible databases that future research can use it and extend the information around these genomes and the natural occurrences of this. Now, during my career, I had a part where I was heavily involved in foodborne pathogens. Now, for those of you who don't know, South Africa actually had one of the worst listeriosis outbreaks recorded ever, which led to the death of approximately 250 people. Now, what we were trying to do here is to use next generation sequencing to inform government consumers about the presence and the pathogenicity of microorganisms on foodstuff. And we were able to do this using whole genome sequencing. Now, this gave us the best resolution, and it was extremely informative in seeing where the outbreak came from, and it enabled us to sequence in excess of 1,000 isolates, which we were then able to contribute back to public databases and continue expanding this foodborne pathogen surveillance network. Now, this is a very important aspect in microorganisms, in consumer safety, and in databases and data in general, is where we are able to sequence isolates and then deposit the genomic sequences into publicly available databases that we are able to compare new samples with and extract relevant and critical information. So let's move on a bit to microbiomes, which is currently where a lot of my work is happening. So in essence, we've got shotgun sequencing where we are able to determine the taxonomic profile and the functional profile, or we can use amplicon or targeted sequencing approaches, which would enable us to create a taxonomic profile, which may lead to the prediction of a functional profile. So in essence, what we're doing when we're looking at metagenomics and microbiomes is we are reconstructing or building puzzles which consist of various different organisms. So we're building a puzzle which would display a community. Now, microbiomes are critical and they form one of the key components of One Health, where human, environment, and animal health is all connected. Now, this health of the ecosystem relies heavily on the microbial component, and the microbiome has even been labeled the second genome. So we really need to get a better understanding of this One Health conduit and reservoir. And this is where a lot of my work currently is. So regarding soil microbiomes, we were very interested in the presence of antibiotic resistance genes, and we had samples from the central Namib. This is a hyper-arid desert on the southwest coast of Africa with minimal anthropogenic influences. It's mostly the scientists going there, dirtying up the place. For this, we use metagenomics to search for antibiotic resistance genes in these soils, and we could pick up a clinically relevant plasmid-borne ARG. This contained a metal resistance gene, and it was resistant to third and fourth generation antibiotics. And this indicates the contribution of microbiomes to the environment and One Health. Then we had a look at some plant microbiomes, and here we're looking at the influence of pathogens in sorghum. Now, sorghum recombinant inbred lines were allowed to be infected naturally by pathogens, and they were grouped into resistant, moderately resistant, susceptible, and highly susceptible groups. There was a clear association between the plant health and the microbial community structure, where we could see a distinct fungal and bacterial patterns for these. Plant growth promoting organisms were associated with the resistant and moderately resistant groups, and then a high diversity of the fungal and bacterial taxa were found in asymptomatic recombinant inbred lines. Then we moved on to some animal microbiomes, and here we were actually interested in looking at the health, the microbiome of sheep, and then the Barbus bollworm, which is a parasite which is found within the gut of the sheep. Now, this indicates also the potential nutritional importance of the microbiome to the worms inside the sheep. And we found that the male worm gut microbiome was more diverse than that of the female. There was also a significant difference between the host and the parasite gut microbiome, even though the parasite spends the majority of their life cycle inside the host. So this indicates a unique microbiome from its host, which could be important for its survival. This further leads to questions surrounding microbiomes and the control of the gastrointestinal parasites in livestock. We further expanded this to look at the gut microbiomes across a vast multitude of different species, and we were really interested in looking at antimicrobial resistance and the occurrence of antimicrobial resistance in different livestock species. And we could clearly see that between the different animals, there was a vast difference in the presence of antimicrobial resistance. Obviously, we had to have a look at some human gut microbiomes, and currently we're busy wrapping up on this study where we are looking at the gut microbiomes of rural and urban South Africans. This is focused on answering questions along the effect of urbanization on the gut health of individuals. So what we could see is that in the rural cohort, we had a higher alpha diversity when compared to the urban group. And we further expanded this into community or network analysis, where we found the urbanized group to have a more disjoint community structure. In the rural individuals, the communities were closely linked to each other, indicating a cohesion and a sense of working together. Beta diversity and differential abundance analysis further indicated that there was a great difference between the guts of the urban and the rural individuals, which alludes to the fact that urbanization and a westernized diet greatly influences the gut health of people. During COVID, obviously everyone was jumping on the bandwagon of SARS-CoV-2, and we used metagenomics to have a look at the presence of SARS-CoV-2 in wastewater. For this in particular, we used metatranscriptomics, and using enough sequencing or throwing enough data at it, we were able to positively identify different variants in the wastewater samples, and we were able to track these across different time periods and relate it to the number of active SARS-CoV-2 cases. This further allowed us to reconstruct MAGs, or metagenome assemblable genomes, from the wastewater, and this gives us the opportunity to have a look at the genomes present in the wastewater as a proxy for human gut health. So what I want to talk about now actually is probably what a large portion of this data fund is about. It's about public data. Again, coming back to the fact that I'm a bioinformatician, so you would not find me in the field. I wouldn't really be out there collecting information. But what I do is I sit behind the computer and I collect information off the internet. Now, some of the greatest repositories I found was the NCBI Short Read Archive or the European Nucleotide Archive. Now, what is great about this is that you can supplement any of your data sets with data available, or you could create a hypothesis based on the publicly available data. I further enjoy a database called Progenomes, which is very nice about this database is that it groups organisms or samples according to the biome or the location it was found in. Again, to illustrate where we are moving with regards to data and the sharing of data and the availability of the data would be projects like the Microbe Atlas. Now, this is a fantastic website where you are able to collect information being sequencing data from all over the world, including categorizing your search for certain locations, biomes, and certain habitats. Again, all of this data is publicly available and free for anybody to download. Another one which I regularly use is the EBI Magnify portal. And this again allows you to search for publicly available data without having to pay or carry the cost of sequencing your own data. So what I'm not saying is that new data or new sequencing projects are not necessary, but that we do have a wealth of data out there which is still unanalyzed or not analyzed to its full potential. So maybe coming to the last section here is I want to give you an example of how we are currently using some of this public data to generate additional knowledge. So this is a very, very nice tool, which is based on the NCBI, and it's called the Microbiomes website. And this is actually where the majority of the pathogens that are detected are hosted, and it includes information on the virulence and the antimicrobial resistance presence in these acids. So what we did is we extracted the 15, what it was roughly 50 million records off the NCBI, and we were able to group the occurrence of antimicrobial resistance according to sources such as agriculture, clinical, human, environment, and we are able then to calculate abundances. All of this by just using publicly available data. This further allows us to group organisms into the escape pathogens and the non-escape pathogens. And from this, we could clearly see that there was a very high incidence or occurrence of antimicrobial resistance, in particular in the escape pathogen group. It further allows us to detect which antimicrobials are present in which World Health Organization groupings. And this allows us for additional insight into where antimicrobial resistant burdens will be happening. So in conclusion, I want to come back to the African part of this datathon, is that what we need African data and African microbiome data, and we need to supplement this currently deficit area here with African microbiomes. The more African microbiomes we produce and submit and make publicly available, the more research we are able to do, and the greater we will see the need of African microbiomes and the diversity which is held within African microbiomes. So maybe then for the last slide, as a bioinformatician, I think the most critical component of a datathon is the people and capacitating the researchers in using the data. In essence, having the data available will be nothing without having the bioinformaticians or the computational biologists adequately trained to use and analyze this data. So in essence, that's my story. Thank you very much.